Okay, for this video we are going to do a quick review on the anatomy of the renal system. We are not going to go anywhere near in depth as what we did last semester. So the first things on this one, let's look at the gross anatomy, the macroscopic structures, if you will, of the kidney. So let's look at this, if I can get my things going here. Okay, so if we look at starting with the gross anatomy on this picture, let's first break down the kidneys. On the outside, you're going to have a capsule, a renal capsule that kind of, let's see, do I have a, yeah, it's color. It's just kind of encases the kidneys. And then once we look at the kidney's broken down in your two primary regions. You're going to be looking at the cortex. Let me do a different color here, something that will stand out. Let's try green. Okay, so the cortex is going to be this region. Okay, and you can see the difference in color on those. So the cortex is the outer portion. When we talked about the adrenal gland, there's a cortex and a medulla as well. Cortex is always referring to the outer zone of this. The medulla, renal medulla, is going to be this region, okay? So it's all going to be all throughout this area. That's going to be the renal medulla. Now the renal medulla has a separate structure separate structures contained within that. So let me do, I don't know, yellow might be able to work. Yeah. So this is the renal pyramids, okay? Renal pyramids are going to be essential for a specific type of a nephron. We'll get into that. And this is going to be dealing where concentrating of the urine is going to occur, or concentrating the salt concentration. At the very tip of this region is the renal papilla. Okay, and from the renal papilla, then we get into, let me go back to the green, then we get into the minor calyces in this area. Minor calyces, so we have one here, we have one here, we have one here. Those join together to form a major calyces. Major calyces join together, so kind of eliminate the blood vessels here. So we're looking at the major calyces joining together to form the renal pelvis. And then the renal pelvis drains into the ureter, and then the ureter leads to the bladder, and then out. Okay. So the key structures is going to be the cortex. That's something that you're going to have to be aware of, because when we talk about nephrons, the nephrons are going to be located in the cortex or the medulla, and there's going to be a couple different type of nephrons. But the process of urine formation, as it goes through the nephrons and the collecting ducts, will end up draining, or it drains through the renal papilla, goes into the minor calyces, major calyces, pelvis, and then ureter to the bladder. Okay, so that is your basic gross anatomy review of anatomy. Now let's look at something else here on this one. Okay, on this one. So now let's look at more of the microscopic or functional anatomy of the kidney. Okay. The functional unit of the kidney is the nephron, and that's what we see in this picture right now. That is your nephron, and the nephron is made up of a few different regions. The first one that we're going to be looking at is the renal corpuscle. Okay, the renal corpuscle is actually made up of two substructures. There's the glomerulus. So the renal corpuscle, okay, so this is in green. So this is the renal corpuscle, and it's also referred to as Bowman's capsule. Okay, so let me just do renal corpuscle. Yeah, I'm not paying attention to what I'm writing. P-U-S-C-L-E, renal corpuscle, okay? And the renal corpuscle is made up of the glomerulus, and the glomerulus are all these little capillaries on the inside, okay? So those are glomerular capillaries. And this is where we're going to start when we really get into the physiology of this, because this is where we start forming filtrate. So the glomerular capillaries come off of an afferent and an efferent, and again, I am intentionally mispronouncing these words because afferent and efferent sound too similar. So we'll go with, here it says the renal artery, but we want more specifically going to be, so this is going to be efferent arteriole, and then this one leading into the glomerular capillaries is going to be afferent arteriole. Okay, so those are the two blood vessels leading into the renal corpuscle that feeds the glomerulus, afferent and efferent arterioles. Notice that they're both arterioles, not a venule, okay, because there's no gas exchange occurring in this area. Then once we lead off of the renal corpuscle, let me just change my colors here. Let's just go back to black on this one. Okay, so now as we have the whole structure here is the nephron, then we start going in this direction. You can see it's all these loops kind of going all which way, okay? That's called your proximal convoluted tubule, or PCT. Is it listed on here? Proximal convoluted tubule. Okay, it's also PCT. So I'll be abbreviating PCT frequently on this one. The proximal convoluted tubule leads into the nephron loop, also known as the loop of Henle. So nephron loop. So those two are the same, okay? And that's this structure coming down here, going back up, all right? Then we get into this other structure, then that's known as your distal convoluted tubule, which we abbreviate DCT, okay? So proximal convoluted tubule, nephron loop, distal convoluted tubule. The reason it comes up with proximal or distal, it's what's closer to, if you were to follow the tubular fluid, the secretions, which is closer to the renal corpuscle. Proximal is going to be closer to that, distal is going to be further away. So you can see your proximal tubule, it comes right off of this. Distal starts way over in this area. Okay. Now there's two different types of nephrons. If you were to look at this picture, you can see we have a set color like a yellowish and then an orange color down here. What this is referring to in this case is there's two different types of nephrons. There are cortical nephrons, those nephrons that are found within the renal cortex, which is going to be this area. Okay, so that's going to be your cortical nephrons. And those occupy about 85% of the total nephrons in your body. So those are your primary ones that are filtering the blood and doing a lot of that work. The remaining 15% are called juxtamedullary or juxtamedullary nephron. So that is not written out here. So let me abbreviate that. So we have cortical nephrons, which are about 85% of the total nephrons, and juxtamedullary are about 15% of the total nephrons. And if you were to go back and look at this area, so the cortical nephrons are going to reside in the cortex, juxtamedullary are going to reside kind of in this area, and they dip down in here. So the nephron loop dips down in here, and your proximal and distal tubes are in the cortex. So proximal distal convoluted tubule, the renal corpuscle reside in the cortex, and the nephron loop dips down into the renal pyramid, kind of directing towards the renal papilla. That's the direction that they're kind of going towards. And those are called juxta medullary nephrons. And these nephrons are responsible for salt concentration. This is going to be relevant when we talk about the counter transport multiplier, countercurrent transport multiplier. This is for concentrating the urine, okay? So that's going to be a really important component of this one. Next up, we're going to have collecting tubules. So proximal convoluted tubule, nephron loop, distal convoluted tubule, and basically this area, okay? This area is called the collecting tubule, okay? Collecting tubules lead into the collecting duct. That's this structure. So this is a collecting tubule, that's a collecting tubule, that's a collecting tubule, and all of this is your collecting duct, okay? Both the collecting tubule, I'm going to get this right, both the collecting tubules and collecting ducts respond to antidiuretic hormones. So collecting tubules and duct respond antidiuretic hormone. Okay, and once we get into more detail about ADH, you have a cursory knowledge of that right now, and you know that ADH decreases urine output. So these are the regions that ADH has an impact on or has an effect on, where we reabsorb some of that water. Okay, and then as this goes down, it leads into the papillary duct. Let's just go back over here. So the collecting ducts head down this way, and so this is going to be the papillary duct area. And then the papillary duct drains the urine into the minor calyces, then to the major, and so forth. Okay, so that is your basic outline. Let me just cover a couple other things. I'm missing the juxtamedullary apparatus. This is going to be another important structure we're going to focus in on. The picture here just shows the renal corpuscle on this side, then it goes into the PCT, nephron loop, DCT, and it seems to go further away from this renal corpuscle. It really doesn't work quite like that. This is more of an accurate representation, although not as convoluted as you would like. But you can see we have our proximal convoluted tubule in this area. Then we go down the nephron loop, descending limb, ascending limb. And then as we come into here, you can see it is in between the afferent and efferent arteriole. So afferent, efferent, and those are arterials. So that's a key thing, because this area, what we're doing here, is going to be down in this picture. So afferent arteriole, efferent arteriole down here. This is part of the junction of the distal convoluted tubule and the ascending limb of the nephron loop, okay? And there's specialized cells in this area, okay? And those cells make up the juxtamedullary apparatus and juxtaglomerular apparatus. Those are going to be very important in other things that we're going to be talking about. Now, of all the distal convoluted tubules, the collecting tubules, and the collecting ducts have two special types of cells. So I tell you what, let me go back this way. Just get some fresh area where I can write on. Okay, so distal convoluted tubule, collecting tubule, and collecting duct have specialized cells. Two specialized cells. Okay? So the two cells, the first one is going to be called principal cells. And principal cells, there's two types. Well, principal cells, well, first of all, they bind with both ADH and aldosterone. Okay, those names should be familiar with you because we did cover them last semester. We will cover them again. But the principal cells bind with both ADH and aldosterone. And the last, or the second type of cell, are intercalated cells. And intercalated cells regulate urine and blood pH. Okay? And of the intercalated cells, there's going to be two types of cells. There's type A and type, take a guess, type B. There you go. Type A cells are responsible for eliminating, let me just get rid of that, are responsible for eliminating hydrogen ions, and type B, bicarbonate ions. Okay, so if you need to adjust your pH, remember 735 to 745 of the blood, you either retain or eliminate hydrogen ions, or you retain or eliminate bicarbonate. Okay, so those are the different types of cells that we're going to be looking at, principal and intercalated. So that is your a really quick overview of the anatomy. And we're going to go through all these in various degrees of detail. Next up, we are going to look at the formation of glomerular filtrate. Thank you.